Topic of the lecture today is pathogenesis of cancer. So regarding pathogenesis of cancer, we have to know how does the cancer develop. And in order to know that, we have to know some terminologies before we get into the pathogenesis of cancer so that you might understand what we're talking about. First of all, let's talk about deletion. What is deletion? Before we dive into deletion, we know that all the body structure is regulated by DNA structures. All the cells in the body, all the small cells which are even microscopic, they make up, they form a group and they form a tissue, they form an organ, and it goes on and on and on. It forms the whole body. So actually it's the DNA that codes the whole body. So whenever there's any kind of damage to the DNA, it results in devastating results. Maybe a very minute part of DNA regulates your brain functions. Very minute part of DNA regulates your hormonal system. So any kind of pathology, any kind of termination or deletion in that part of the DNA might lead to devastating results. So let's talk about deletion. What is deletion? Deletion means if part of the DNA, if any nucleotide is deleted from the DNA, is not there anymore. So if that part of the DNA is not there anymore, maybe it's the non-functional part of the DNA, maybe it's not dominant and you won't even know that you have that missing part of the DNA. But in most of the cases, that DNA part which is deleted, that nucleotide that is deleted, contributes to major functions. And you lead to formation of those pathologies which result due to the deletion of that nucleotide. So the term deletion comes from that if any nucleotide or the group of nucleotides or any base pair is deleted. Next on, there's translocation. What is translocation? Trans means inter, location means if the location of one nucleotide is changed with the location of another nucleotide. For example, there's a nucleotide which forms melanin in your body. It's responsible for melatonin. It's activated, but somehow it got translocated with another gene which is not activated at all. So translocation means if one part of the DNA is translocated with another part of the DNA. Maybe that part of the DNA was not functional. That part of the DNA was not functional at all. But now when it's shifted to that part of the DNA, that place of the DNA which is activated, now it will lead to results according to its base, according to its code. Next on, we have amplifications. What do you mean by amplifications? Amplification means if part of one DNA, if one part, specific part of the DNA is amplified, like there are multiple copies of that part of the DNA. It was supposed to be like 3 copies, or it was supposed to be in 3 places, and among them 2 must be activated, or one of them should be activated. Now, because of the amplification process, multiple copies of that part of the DNA has been produced, and it will result to maximum impact that part of the DNA can have on the body. Next on, we have depression. For example, this can also, we can correlate depression with translocation. That, for example, if this part of the DNA is activated, but it got translocated with this part of the DNA. Now it was active, but it was not. Now when it's shifted here, it's activated now. But if this part of the DNA is shifted here, which was non-active, it's depressed. Now its functions won't be visible. Now its coding is not of any use because this part of the DNA was not supposed to be active. This is one example. In some cases, some parts of the DNA, some parts of the chromosome, some nucleotides go repressed because of some mutations. And last but not the least, point mutation. Point mutation means there's a specific point, there's a specific nucleotide which has been mutated due to anything. So these mutations, these changes can occur due to many things. These changes you can inherit them, you can get them from carcinogens like chemicals or arsenic or something like that, or you can even develop them over the time. Over the time period, you develop these mutations. So If DNA is being mutated, why the body is not taking any action? That's what we're here to learn right now. So before we learn how the DNA is mutated, how a whole body goes into that phase of the cycle in which cancer propagates, we have to know the normal physiology of the cell cycle. So the normal processing of the cell cycles are, these are the phases of the cell cycle, normal cell cycle phases. Actually, there are G1, S, G2, and M. But there's this one stage called G0. So actually, there are 4 actual stages of cell cycle, and then there's this resting phase, which is called G0. G1 mean gap. G1 mean first gap, or gap 1 phase of the cell cycle. It's that phase of the cell cycle in which the cell becomes more in size, it enlarges, it increases its size, and with its size, it does not multiplicate, it does not replicate. It's just increasing in size. It's making sure that everything is in place. It's making sure that before we get on to DNA synthesis, all the DNA is in place. All the chromosomes are in the right place before we go on to DNA synthesis. Before we make a copy of it, we have to be sure that everything is in place. There's no mutation whatsoever, and then we can move on to S phase. There's this restriction point. At this restriction point, major physiological things are happening. This is the phase in which the cell determines that whether it has to move on or go into the resting cycle, go into the resting phase. Maybe the cell is not ready. It goes into a limbo. It needs more time for preparing that. It needs more time for getting into S stage of the cell cycle. So this restriction point is very important. This is actually a checkpoint. For example, you're going for a trip, and then at the end of the preparation, you stop your mind for a little bit and think about the things you need for that trip. And if you're forgetting something, you might need to get that. So it's just like that for restriction point, that at this point of the cell cycle, the cell stops. The cell checks whether it has all the infirmary it needs, whether it has all the needs satisfied or not. Once it knows that the cell is ready for DNA synthesis, it moves on to S phase. In S phase, DNA is synthesized. When DNA is synthesized, all the body structures which were waiting for the replication, waiting for the multiplication, will now move on. So in S phase, DNA synthesis is happening. Then comes the G2 phase. In G2 phase, G2 phase is actually gap 2 phase. So in G2 phase, the cell elongates its size. The cell enlarges in size. So when the cell enlarges in size, what is it trying to do? In G1, it was enlarging in size, and it was getting to the point that it has all the things it needs. In G2 phase, it's actually going to go through mitosis. So it's actually elongating in size so that mitosis can occur very smoothly. Then comes the mitosis phase. At this point, there's another restriction. You can see there's another restriction. So at this point, the cell checks, okay, we are going to go through mitosis. Are we ready for this or not? Do we have that cell structure that will go through mitosis and will lead to 2 cells formation or not? If it's not, it's stopped right here. Otherwise, it goes through mitosis. Cells are divided. Now one cell has divided into 2 cells, and then it goes on to G1 stage. Now, what is this G0 stage? G0 stage is actually the stage of the cell at which the cell decides, I'm not gonna divide anymore. I'm not gonna multiplicate anymore. I'm just gonna stay the way I am. So it goes through all these stages, but it does not go through replication and mitosis and synthesis. So what's the basic example of G0 stage? Brain cells. They do not go through mitosis again and again once they're mature. That's why if there's any brain injury, if there's any ischemia, if there's any hemorrhage, what happens? The effects are lifelong because it's not going to multiplicate and regenerate the cells. It's going to stay that way. So brain cells spend their whole lifetime after maturation into G0 stage. But all of these stages can also result into G0 stage in which the cell is not ready. Maybe the cell goes into a limbo and cell is not ready to multiplicate. Cell is not there to divide itself. So all of these stages, you have to remember these stages and these checkpoints in order to understand what is about to come. So we talked about normal cell cycle, right? So in normal cell cycle, genetic changes in cell causes cancer. Now, if any of those stages are affected, it will lead to the formation of cancer. Because if DNA synthesis is not happening properly, if there's foreign DNA, if there's a deletion of the gene, if there's insertion of some gene, if there's translocation, if any of those things happen to the cell cycle, the DNA copies that we made for that cell are not of the original nature. So what's it going to do? It's going to multiplicate with that altered DNA and it will lead to the formation of cancer. So genes define amino acids sequence of proteins. So if there's gene mutation, okay, the gene is mutated, the DNA is mutated. Now once the DNA and gene is mutated, what's it going to do? What's the function of DNA of the cell? The function of the DNA of the cell is to make amino acids. What amino acids does? They group together and they form proteins. And if the amino acids are not normal, if amino acids are of pathogenic nature, they're going to lead to aberrant proteins, not normal. So it will lead to, one way or another, it will lead to somatic and acquired DNA alterations. Somatic cells will be altered, DNA alterations are already happening. So due to those DNA mutations that occurred right here, different proteins are produced which are not of normal nature, which are not normal in our body, and it will lead to DNA mutations are already happening. Now somatic alterations are happening. Due to that code of the DNA, different kind of structures are being produced which are not of normal nature. And it will lead to polymorphisms and different structures which we do not want in our body. Different pathological cells which we do not want in our body. And this term polymorphisms, these are actually variations in DNA sequence. Now those variations are happening again and again at some time, at some level, that DNA which is mutated already will go through another mutation and it will go on and on. That's why the cure of the cancer has not been found yet. It has not been there has been no cure for it because mutations are happening constantly. So what kind of mutation are we talking about? What should we deal with? It's mutating all the time. So if any minute change happens inside the DNA, it will lead to devastating effects in the somatic structures, in the DNA structures, and at the end it will lead to cancer formation. Next now we have cyclins. While reading the text, you might be wondering what cyclins are. So we learned about the cell cycle. So there must be something regulating the cell cycle. One thing we have been studying again and again and again is that there's regulation to every system inside the body. There's not a single system left unregulated. Otherwise, it will go haywire. And we won't even know where this thing is coming from. So the cell cycle is regulated by two kind of things. One, which gives it a positive regulation, and those which give it a negative regulation. Positive regulators speed up the cell cycle. They provide the cell cycle with the things it needs, and the cell cycle continues the way it is supposed to. And then there are substances, there are proteins which inhibit the whole system, which stop the whole system. Example of that is P53. We're going to talk about it later. But first, let's talk about cyclins. So cyclins are actually cell cycle regulators, and they're positive cell cycle regulators. They're gonna help the cell cycle move forward. There are further subtypes of cell cyclins, which are cyclin D, cyclin E, cyclin A, and cyclin B. You can remember them by A, B, D, and E. So we have learned about these stages, that there's G1 phase which goes into S phase for DNA synthesis and goes into G2 phase, in which the cell elongates and enlarges in size and gets ready to go through mitosis, and then mitosis comes along. So which of these cyclins are there for the whole cell cycle, and which of these cyclins are there for just a brief moment? In this slide, this depiction of them, that cyclin D, you can see the cyclin D starts from G1 phase and it goes throughout the cell cycle till the end of mitosis. On the other hand, cyclin E starts from the middle of the G1 phase, ends in the S phase. Then comes cyclin A, which also starts in G1 phase but ends in the very early stage of mitosis. And so then comes cyclin B, which starts in the S phase and ends in the middle of mitosis. What's the importance? Why are they there? They're the positive regulators of the cell cycle. They have to ensure that cell has all its infirmary to move forward in the direction of replication, to move forward in the direction of mitosis. You can see all of them are ending in mitosis, some of them even in the early stages, and some of them even in the later stages. So from this depiction, you can understand what's the most important cyclin. Cyclin D, because it's present throughout the cell cycle. And then there are further subtypes of it. You will learn about CDK. They are cyclin-dependent kinases which help regulate the cycle. Now, CDK is present here. It's cyclin-dependent kinase. One of these cyclins will come in, attach itself with the CDK, and then the action of the cyclin will start. So cyclin-dependent kinases will carry cyclin, either one of them, and the functions of the cell cycle will propagate, will enhance, will move forward. So before we move on to knowing more about the cell cycle and at which stage which cyclin is acting, we have to know about the oxidant entity. Now, what is oxidant? Oxidant is anything which is free radical inside the body. So they affect the body, they affect the cells, they even mutate them in some cases. So they have a devastating effect on the cell cycle. So whenever there's lipid peroxidation, carbonyls MAD and 4-HNE will be produced. Now, what's their function? They cause a DNA damage. They can either do any kind of damage. They can either do point mutation. They can either do deletion of some nucleotide. One way or another, it leads to DNA damage. And it's because of free radicals that are present, which will lead to ATM and ATR, which are mutations in the gene. And what will happen? P53 will be activated. I told you we're going to talk about P53. Let me tell you that P53 is actually a protein which is present on the chromosome 17. Now, chromosome 17, why is it called 53? 53 is actually its weight. Kilowongton is the unit of that weight. Now, protein which is present on chromosome 17 is 53 kilowongton. So that's why it's called P53. Now, P53 has multiple actions, multiple functions. P53 is negative regulator of the cell cycle. We talked about the positive regulators of the cell cycle, which were cyclins. Now we're talking about the negative regulators of the cell cycle, which is P53. One of them is P53. So whenever there's damage to the cell, whenever there's mutation in the DNA of the cell, whenever there's any kind of DNA damage, amount of P53 rises in the cell. When amount of P53 rises in the cell, it stops the reaction right there. It does not let the cell go further into the stage. At which stage you might be wondering that P53 is acting? It's acting at G1 stage. So at the restriction point, at the checkpoint of G1 stage, P53 is present. Now P53 can either be in short amount and maybe it's uh, destroyed in some way. We're going to talk about later that how it's deactivated or destroyed, and then the cell moves into S stage. Now P53 is actually kind of an inspector which comes when there's a problem. When there's a problem, what happens? What happens is that P53 increases in amount and it stops the whole system. P53 knows that there's a damage inside the DNA, so it won't let the stage go from G1, it won't let the cell go from G1 to S phase. Because in S phase we have DNA synthesis. If there's any damage to it, the DNA produced in that S phase will be mutated, will be not normal, will not be normal at all. So it's actually stopping the production of abnormal DNA. It's stopping the production of mutated DNA. At this stage, we have CHK1 and CHK2 regulating the production of it. It also produced P53, phosphorylated. You can see the P alongside it is phosphorylated. There's two types of P53. It's either phosphorylated, it's either dephosphorylated. We're going to talk about it in P53 section. So when P53 is produced, because of both of these, what happens is P21 is produced. P21 is a protein which P53 produces. Now P21 is actually antagonist of P53. What happens is that P21 won't let the cycle go further. P21 will stop the cycle right there. P21 does not negate P53. It's actually going to negate what? Cyclins. It acts on the cyclins. It stops their functioning. And as a result, cell cycle stops because cyclins have been stopped due to P21 action, protein 21 action. And cell cycle goes into arrest and DNA damage repair system is activated. I told you P53 has multiple functions. One of them functions is repairing of the damaged DNA. When the cell cycle goes into arrest, it's at the checkpoint, it's not going to move forward to synthesis stage. So the cell has now time. The cell has now time to make up for the mistake it made. Now cell has time so that the DNA which is damaged can be repaired. And when that damage has been repaired, it can be sent, it can move on to S phase. Now the DNA is fine. We can go into S phase. We can synthesize the DNA. Now the cell will be functioning normally. But at this point, P53 comes in to stop us from oxidant injury. Due to P53, one thing that happened was cell cycle arrest and DNA damage repair. But what if the DNA is beyond repair? The damage is too much. P53 cannot make up for the whole damage. Maybe there's deletion of a gene. Maybe there's translocation and P53 cannot comprehend with that kind of injury. It produces these proteins, which is called FASR, BAC, SPUMA, and NOXA. They are apoptotic. They put the cell through apoptosis. The cell goes for suicide. The cell dies. Before it gives birth to abnormal DNA, before it gives birth to abnormal kind of DNA synthesis, the cell goes through apoptosis. That's what P53 is present in our body for. So we were learning about cyclins, the positive cell cycle regulators. The cyclins and cyclin-dependent kinases affect the cell in this way. We talked about D cyclins, which will go throughout the stage. We talked about E cyclins, which will start here and end in S phase. We talked about cyclin A, which will start in S phase and go through the whole stage and end at the middle of mitosis. And then we talked about cyclin B, which will start and end right here. So the function of the cyclin is positive regulation of the cells. Positive regulation of the cell cycle. It provides you with all the armor which is needed for the cell to move forward in the direction of mitosis. Now, how these cyclin-dependent kinases, this CDKs mean cyclin-dependent kinases, that structure which holds the cyclins. So, how are they regulated? Okay, they're present. How are we to increase or decrease their amount according to the situation? There are growth factors present and cytokines present, which are very important in determining whether cyclin and cyclin dependent kinases are needed inside the cell. So when growth factors and cytokines act on it, the gene expressions promoting cell divisions will be produced. What are those genes? Proto-oncogenes. Proto-oncogenes are actually the genes which promote the growth, which give rise to the new cells. So when proto-oncogenes are produced, what proto-oncogenes are going to do, they're going to promote the cell division. They're going to promote the multiplication and the division of the cells. Next on, the production of D&E cyclins will happen. And what D&E cyclins are going to do? They're going to get attached to cyclin-dependent kinases 2, 4, and 6. We talked about which cyclins are most important. We talked about D basically and E in some cases. So which cyclin-dependent kinases are going to be important? If D cyclin is going to be important, the cyclin-dependent kinase which is going to hold it must be important. So cyclin-dependent kinase 2, 4, and 6 are very important. On the other hand, cyclin A is going to lead to cyclin-dependent kinase 2 and cyclin-dependent kinase 1. And cyclin B will also lead to cyclin-dependent kinases. And these are activated in this manner. So this is how cyclins and cyclin-dependent kinases are activated due to growth factors, cytokines, and then proto-oncogenes. Now, if there is any damage, if there's any DNA damage or errors in replication or any kind of oxidant injury, we talked about what will be produced? P53. So P53 will be produced, which will halt the cell cycle, which will stop the cell cycle in its route before going to S phase, before synthesis of DNA. So P53 will lead to all of these changes, which will include CKIs, INK, CYP, KIP, and RB families, retinoblastoma families. Now these are always you have to remember, these are also the negative regulators of the cell cycle. They're going to halt the cell cycle. So this will try its best to halt the cell cycle, and if the cell cycle is being halted, it's going to halt this activation too, so that these are not produced, or if they are produced, all of these negative regulators will stop their production or cancel their presence somehow, and it will stop the production of or stop the progression of the cell cycle from G1 phase to S phase. Next on, we have CDK inhibitors. Now, cyclin-dependent kinases that we learned that they are going to take cyclins and they're going to act on it, and they're the positive regulators. Now, how they are inhibited? How a cyclin-dependent kinase is inhibited? First of all, it's going to be INK4 proteins. They are the inhibitors of CDK, cyclin-dependent kinases. They bind and inhibit CDKs 4 and 6. We talked about which are the most important cyclin-dependent kinases 4, 6, and 2. So if they're going to bind and inhibit CDK4 and 6, what's going to happen? That CDK4 or that CDK6, which was going to take cyclin D or cyclin E, is now inhibited. So cyclin D, cyclin E won't be activated. The cell cycle won't continue because cell has a need for the production and the presence of cyclins. So if this is inhibited, the major cyclin is inhibited. The major cyclin is of no use. So cell cycle halts in that process. Next protein is CIP and KIP proteins. What they're going to do, they're going to bind and strongly block CDK2. Now CDK2 was being produced by, yes, also cyclin D and E. So if 2, 4, 6, all of these are blocked, cell cycle won't continue. So this also has a way of getting back to cyclin D and E. Next on, we have RB family members, retinoblastoma family members. Now, this is the most important one of the negative regulators. Where is it acting? Exactly at G1, exactly at the resting stage of the G1, so that the cell synthesis, the DNA synthesis won't continue. It also targets cyclin-dependent kinases too. So all of these CDK inhibitors are going to target either 2, 4, or 6, which are important for cyclin D and cyclin E. So we talked about RB families, that it's going to affect basically CDK2. So how is it acting? How is it forming? How is it involved inside the whole cell cycle? So we're going to talk about retinoblastoma protein and its family. So what retinoblastoma is? It's actually present on chromosome number 13, and it codes for retinoblastoma proteins. And retinoblastoma proteins are present in the G1 stage. They're attached to E2F factors. Now, what is E2F factors? What is E2F basically? Cell won't start the transcription process. Cell won't start the DNA synthesis unless E2F is attached to it. E2F is attached to the hereditary structures. E2F is attached to the chromosome. So if E2F is attached with retinoblastoma and retinoblastoma won't let it go, DNA synthesis won't even start because it needs E2F for the proper transcription or for the synthesis of the DNA. So if retinoblastoma is attached to E2F, it means it's the basic inhibitory regulator that we have. If it won't let it go, transcription won't start. DNA synthesis won't happen. Mutation won't be continued in the next cells. So activated cyclins D, E, and then we talked about cyclin-dependent kinases 2, 4, 6. They're going to phosphorylate the retinoblastoma. When they phosphorylate the retinoblastoma, it lets go of the E2F. Normally, retinoblastoma is hypophosphorylated. Hypophosphorylated means it contains less phosphorylation. It has less phosphorylation. Less phosphate groups are attached. When more and more phosphate groups are attached, when it becomes hyperphosphorylated, what happens? It lets go of the E2F. It has its capacity being used by phosphate groups. So these activated cyclins, D and E, which are being carried by cyclin-dependent kinase 2, 4, and 6, they're going to come in, they're going to phosphorylate the retinoblastoma, and what happens in the result? That retinoblastoma is now phosphated with PO4, and E2F is free to go. Now when E2F is free to go, what is happening? Cell cycle is progressing. E2F is going in, attaching itself to the chromatic structures, and then the process of DNA synthesis starts. So what happens if this phosphorylation doesn't happen? What is phosphorylating this retinoblastoma in order for it to let go of E2F? These cyclins. And we talked about these inhibitors. If they block CDK2, 4, 6 and cyclin D and cyclin E, there will be nothing to phosphorylate the retinoblastoma. And if retinoblastoma is not phosphorylated, it won't let go of the E2F. And if it won't let go of the E2F, transcription process won't even start. DNA synthesis won't even start. Next on, we have role of cyclins and cyclin-dependent kinases. We have studied like 2 to 3 uses of them, which were that they phosphorylate the retinoblastoma. They are positive regulators of the cell cycle. Now, let's see what's the other role of the cyclin and cyclin-dependent kinases. What happens is that when there's DNA damage, any kind of DNA damage, it can be due to carcinogens, it can be due to chemicals, it can be due to even UV lights. If there's any kind of DNA damage, ataxia, telangiectasia mutated happens. That's what it's called. It's a terminology you have to remember. What happens is that this phosphate group will be here, and this stalled DNA replication will be seen. DNA replication is now stalled. So what happens is ATM and RAD3 related ATR will be produced. Now it's not normal. It's not going in the direction it's supposed to go. It's not going to synthesis. It's going to halt in the process. So what's going to happen? So this in turn will lead to the production of P53. We know that P53 is going to halt the whole process. It's going to affect the whole process. It won't let it continue. When P53 won't let it continue, what will happen? The whole effect of cyclins will be cancelled. How? Let's learn how. This P53 is going to be attached to it and further move on, and the production of MDM2 happens. Now MDM2 is actually present alongside P53 whenever there is damage happening, whenever the amount of P53 rises. It's there to cancel the effect of P53. It's there that in case the cell has to move on, the cell has corrected its mistake, now MDM2 will start the disintegration of P53 so that cell can move on into the next stage. So MDM2 will go on to the one side and P53 on the other side. And what will P53 do? It will lead to production of protein 21. And this protein 21 is actually there to cancel the effects of cyclins and cyclin-dependent kinases. When cyclin and cyclin-dependent kinases are affected, phosphorylation of retinoblastoma won't happen. When phosphorylation of retinoblastoma won't happen, it won't let go of E2F. And when it won't let go of E2F, the synthesis of the DNA won't even start. And there comes protein 14, which affects in their combination and their dispersion. What happens afterwards? So we have learned about all the stages of cell cycle. Now we know that there's G1 stage, then there's S stage. Between them, there's a checkpoint in G1 stage. At that stage, all of these cyclins and cyclin-dependent kinases and P53 and retinoblastoma, all of them are working together. All of them are making sure that the DNA is not mutated and it goes on intact onto the next stage of DNA synthesis. And then it goes to G2 stage, and where the cell elongates, the cell enlarges and gets ready for multiplication, for mitosis. So what are the landmarks? You know the landmarks already. What happens is restriction point. First of all, restriction point. We just talked about G1 stage, the restriction point at which all of these P53s and retinoblastomas, all of these are working together. So in this, cell crosses the Rubicon. Next on, we have replication checkpoint. Before replication, there's another checkpoint which makes sure the chromosomes are present at their spindles. They're not far away. They're present on the places where they're supposed to be so that allele functioning can happen. So the activation of CHK1 to block mitotic separation comes here if there's any problem present inside that spindle network. Next on, we have spindle integrity checkpoint. Now we talked about all the chromosomes are there, but if the spindles are not ready, if the spindles are not up to the mark, this checkpoint will stop the cell cycle right here, and it is activated by kinetochores if inaccurate segregation has been done somehow due to any kind of mutation. Then we have aura B kinase. Aura B kinase ensures that kinetochore binds effectively. Maybe the kinetochore at this stage are not binding properly. So aura B makes sure that it binds correctly. So these are the landmarks of the cell cycle which make sure that the new cell produced has this equal and same and normal amount of DNA which it was supposed to have. Next on, how P53 is regulated and what is MDM2. We talked about MDM2 and P53, they're antagonists, they're going to cancel each other's effect. Now let's talk in detail that what is happening actually. So MDM2 is present whenever there's increased amount of P53. Now MDM2 is going to attach itself to P53 with the help of ARF. Now when it's attached, now it's there to destroy P53 one way or another. So ubiquitin pathway will come in and P53 degradation will start. Now MDM2 is not degrading. MDM2 is there. MDM2 is not going anywhere because P53 is high. It's going to use the ubiquitin pathway and going to destroy the P53. When do we need that P53 should be destroyed? For example, that there was a mutation and P53 corrected the mutation. We talked about P53 has ability to correct some mistakes inside the DNA. Now that mistake has been corrected, but P53 levels are too high for cell to move on to the DNA synthesis stage. So MDM2 will come into play to reduce the amount of P53 or finish P53 present inside the cell cycle so that cell may continue to proliferate. So MDM2 will attach itself to P53 through ubiquitin pathway. P53 is degraded. Now what happens if there is genotoxic stress? It affects P53. P53 will go into disintegration. It will activate BAX genes. We talked about BAX genes. These are apoptotic genes. We talked about BAX are apoptotic in nature. They're going to kill the cell. They're going to cause a cell to go through suicide or kill itself. And this happens in only those cases in which it's not even possible to repair that kind of damage. And apoptosis will happen. But if there was any chance of it being normalized or corrected, P53 would have corrected itself and then would have gone through degradation, and P53 will be reduced and cell cycle will continue to S stage. So the point to get from this slide is that P53 tries its best to correct the mistake, to correct that alteration made in DNA before. When it corrects itself, it invites MDM2 to kill it. MDM2 will come in, it will cause a degradation of P53, but MDM2 will be there so that it can crush the other P53 present inside the cell cycle, present inside that cell. And if the mutation is too much and there's genotoxic stress is present and even P53 is being mutated, even P53 is going through something, what happens is BAX comes in, BAX, it's called BAX, it comes in and causes a cell to go through apoptosis so that the damaged DNA should not be inherited into the new cells. Now, how P53 and MDM2 are regulated? Whenever there is genotoxic stress, P53 comes into play and these mutations happen. It's phosphorylated. Mutations happen. These genotoxic stress can come into ATR, CHK1, ATM, and CHK2. What happens is that it activates CKI, P21, CIPI. Now, these are actually kind of code forms for these mutations that are happening. So what will happen is that it will give off to GADD45, which leads to DNA repair. That damage which has been done, GADD45 will lead to DNA repair. And when the DNA is repaired, P53 will go away with the help of MDM2. Now, if this one is produced, CK1, P21, CIPI, cell cycle goes into arrest. Cell cycle won't continue until that mistake is being corrected. Now the cyclins come into play, and we have these two things like retinoblastoma, which is phosphorylated, and E2F. We talked about whenever they're attached, it means they're hypophosphorylated. Retinoblastoma is hypophosphorylated. Now cyclins will come into the play. Cyclin will hyperphosphorylate the retinoblastoma. It will lead to E2F going free. Retinoblastoma being hyperphosphorylated, E2F going to the genetic material and causes and start the process of DNA synthesis. Now let's talk about the DNA damage and repair. We have learned all the basics that we need to understand how the DNA is being damaged, how it is being repaired. So let's talk about DNA damage and repair. There's three kinds: DNA alteration, repair process, and the representative tumor types. So what happens if it's single-stranded break? If the break is single-stranded, it's not that much. What happens is that this will be taken care of, this will be repaired somehow. But if it doesn't, what kind of tumor will be produced? So first of all, let's talk about what kind of DNA alterations that we're talking about. We have single-stranded break, we have double-stranded break in which both the strands of the DNA are damaged and broken off. Then we have adduct in which one foreign thing or foreign material or foreign nucleotide or something like that is added to it and it's affecting the adherent part of the chromosome. And next on we have base insertion or deletion, in which we are showing base deletion. This base is deleted. We talked about deletion before. So base insertion or deletion can happen. Maybe it's another base. Maybe, as you can see, the base is deleted here. So any of these can happen. Either it can be base insertion, either it can be base deletion. Then we have base mismatch. You can see there's C and there's T. So the bond won't form. Why? Because we talked about adenosine and T will make a bond. On the other hand, C and G will make a bond. Whenever there's T and C in front of each other, they're not going to make a bond. They cannot make a bond. So base mismatch will happen. It's a mismatch, so they won't connect. So what kind of repairing process that we're talking about now? First of all, if there's a single-stranded break, there's base axion repair. Now it can be taken care by even P53. It's a very simple process. Base axion repair will happen and we'll have a normal DNA afterwards. What happens if it's a double-stranded break? Double-stranded break repair contains non-homologous end joining. Now these ends will be joined. Okay, it's gone. We cannot repair it right now, but these ends will be joined. Now that gene is deleted. Now that part of the gene is deleted. Now once it's deleted, it won't have its consequence on the human body. Now the main functions that it was supposed to do, it won't be doing from now on. So double-stranded break happens. So non-homologous end joining will be the treatment or the repair process that will be happening inside the body. Next on we have adduct. The repairing process that is required for its nucleotide axion repair, in which this whole part will be the nucleotide which is it attached to will be excised and then repaired from end to end. Next on we have base insertion or deletion. Depends that if it's a base deletion or insertion. In both the cases, we're going to go through mismatch repair. If it's insertion, we're going to delete it and do the normal process and try to get the normal functioning and normal nucleotide there. If it's deletion, we're going to try and get that deleted part right into it back again. So the types of tumor which are represented in single-stranded break or double-stranded break are pancreas, breast, and ovary. Now all of these are female tumors. So any kind of single-stranded break is there, double-stranded break is there, and repair has not happened. Repair could not happen. What kind of tumors it will produce? It will produce breast, ovary, and pancreas. Adduct only happens in xeroderma pigmentosum. This is something which can come in your board exam, that what happens if DNA is altered in a way that it's adducted somehow. So xeroderma pigmentosum is the only condition which comes with the adduct. Mismatch repair and base insertion, base deletion, or base mismatch, colon and rectal carcinoma will be the most probable representative tumor type in this kind of DNA alteration. So what are DNA repair pathways? We talked about DNA alterations. We talked about DNA is altered somehow. We repaired it. What's the repair pathway? if even if it's successful, if it's unsuccessful, we talked about which tumors it will produce, which tumors will representate them. But what are the repair pathways? So this is the original sequence. G is with C, T is with A is with T. We talked about G and C will always be in front of each other in T and A. So this is the original sequence. What happens if MMR2 mediators come in, and this is something which happens. Now you can see this very different kind of pathology here, that T, C and A are together, but there's no match being made here because it's not sure it's going to make a match with T, but it's unable to make a match with T because we have 3 of them present right here. It is recognized by MSH2, MSH6, and it's either insertion or deletion. Maybe this place is deleted, maybe it's inserted. In both the ways, it can be recognized. This mutation can be recognized by MSH2 and MSH6. And what will happen, that will recognize it with MSH2 and MSH6, and the body, after recognizing it, will try to repair it. We're going to talk about what kind of repair will it go through. So MMR1 mediators will come in. This was MMR2 mediators. So MMR1 mediators and MMR1 mediators, we have GC, CA, AT. We talked about the C is always going to be with G. It's never going to form bond with A. So we have mismatch here. We had insertion or deletion here in MMR2 mediators, and now in MMR1 mediators, we have this mismatch in which the bond won't form. Now it has been repaired by replacing the C with the T so that the bond may form. And the original sequence is restored in this manner. Same will happen with it. If it's insertion, it will be deleted. If it's deletion, the normal sequence will be inserted, and this is how original sequence will be restored. So this is how DNA repairs itself. These are the repair pathways. So we're going to talk about type of damage. We're going to talk about what's the cause of it. We're going to talk about which kind of repair pathways are helpful in repairing them. First of all, if there's base oxidation, the cause can be mitochondrial reactive oxygen species present inside the cell. It can be because of phagocytic generated oxidants. It can be because of UV and ionizing radiations. It can be because of smoking. So nucleotide excision and repair and base excision repair will happen. What do you mean by nucleotide excision? It means that nucleotide is not there. It does not belong there. So we excise that, and after that we base excise. We excise the base and then we repair it with a normal nucleotide. On the other hand, if there's any other base modifications required, which will be due to chemotherapeutic drugs, which will be due to neutrophilic bactericidal enzymes, which will be due to cigarette smoking and other environmental chemicals like carcinogens, we need only base excision repair. We do not need nucleotide excision repair in this condition. In DNA damage and repair, either it can be additions, which can be caused by errors in transcription or UV light or oxidants, or repair by NER, meaning that repair was not helpful, that repair was faulty. Either it can be single-stranded nicks, which can be due to ROS, ionizing radiations, loss of sugar phosphate bond. If loss of sugar phosphate bond happen, what happens? They'll lose. Any kind of mutation can happen. And it can be repaired by BER and NER, but if any kind of pathology and even their repair, it can lead to DNA damage. But basically additions are repaired by NER, nucleotide excision repair, single-stranded nicks are helped with base excision repair and nucleotide excision repair, and then double-stranded nicks are actually due to arrested DNA replication and ionizing radiations and chemical damage, and it can be repaired by HR and NHEJ, meaning nucleotide excision and histopathological geometric pathways. So in DNA damage and repair, What happens is the causes of DNA damage repair are ionizing radiations and environmental chemicals, which can be any kind of carcinogen like arsenic or mercury or something like that. The causes also are due to arrested DNA replication, and repair pathways can, in repair pathways, Fanconi related repair pathways can affect them in causing the DNA damage, and its basic function of it is the repair. But if the repair is faulty, it can also lead to another kind of mutation. What are the hallmarks of cancer? Unregulated cellular proliferation. In the introductory lecture of neoplasia, we talked about the hallmarks of cancer. It's always going to be unregulated. It's always going to be immortal cells and evasion of PCD. And it's always going to be stimulation of vascular proliferation. It's going to affect the vascular levels. And it's going to invade and metastasize inside your body. So these are the hallmarks of cancer. It's going to be unregulated. It's going to affect the PCD. It's going to go inside of your body, invade and metastasize, and it's going to affect your vascular system. And one and the most important point is that it's going to be monoclonal. Further, it has inactivation of tumor suppressor genes. When tumor suppressor genes are activated, there's nothing to stop the tumor from growing. Next on, we have tumor cell growth and spread. How does it grow and spread? It can either be due to genomic instability, and then it leads to altered epigenetic regulation. When there's epigenetic regulation is altered, it just multiplicates and proliferates, and it does not stop at any level. Next on, we have altered bioenergetics. When the bioenergetics are altered, what's going to happen? Faulty DNA is there. It's going to just multiplicate and make into new tissue, new cells, new bunch of cells. And the immune avoidance and inflammation will happen because immune system fail in comprehending this kind of pathology. So what will happen is that immune avoidance will start happening and inflammation alongside with it. Let's talk about proto-oncogenes. We gave a tutorial of you in the very early slides of proto-oncogenes. Proto-oncogenes are actually growth genes which enhance the growth inside the body. So if any mutation is to happen to proto-oncogenes, what will happen? It will lead to the formation of oncogenes. And oncogenes are cancerous genes. When proto-oncogenes are affected, there's nothing to regulate the growth of the cells. So unregulated growth will happen, that you just talked about in the hallmarks of cancer. That if unregulated growth if the growth is unregulated, cancer will start forming. So proto-oncogenes, if there's translocation or transportation gene moved to new locus or under new controls, normal growth stimulating proteins will be produced in excess. If there's gene amplification, multiple copy of genes are present and normal growth stimulation proteins in excess. If there's point mutation within the control environment, normal growth stimulation protein are produced in excess. And if there's point mutation within gene, hyperactive or degradation resistant proteins will be produced. So due to all of these mutations, more and more proto-oncogenes will be produced. When more and more proto-oncogenes will be produced, or even in some cases oncogenes will be produced after mutation, unregulated cell growth will happen. All of these proteins will be produced, and they'll start the cells to just grow and grow without any regulation, and they will start the formation of a cancerous cell or tissue in some cases. We talked about oncogenes. So cancer causing agents, it can be anything, arsenic or UV lights or oxidants. They affect the proto-oncogenes. This place is destroyed. Oncogenes will be activated. DNA is altered, and when oncogenes will be activated, oncogenes are going to produce massive amount of cells which are not of normal DNA and values, and they're producing the cancer cells in this. Next on, we have proliferation mechanism. We're going to talk about how these signaling paradigm act on the proliferation mechanism and how does this affect in some ways. So as you can see, these are the ligands, these are the receptors that they respond to. So this ligand will only fit in this receptor. It's just like some are steroid receptors, some are different kind of drug receptors. So these receptors are present here, they accept the ligand, and they afterwards start an intermediate signaling. And now, before we get on to this paradigm, we have to know that the pathology or the proto-oncogene that codes for this receptor, that can go mutated and it can convert to oncogene, and the pathology may lie right here, or the pathology may lie right here in the ligand. So, and every proto-oncogene actually codes for growth of a different cell, of a part of the cell. So any mutation in the proto-oncogene will lead to the mutation in these structures, and these structures will not respond in the normal way. But first we have to know what the normal way is, right? So this ligand will fit in this receptor, this in this receptor, and you can see this in this receptor. On the other hand, steroids don't need a receptor, they just get in, they just cross the barrier. So all of these will lead to an intermediate signaling. So does this receptor, so does this receptor, and so does this receptor. But this receptor goes to GTP related. Those receptors, those elements which require energy for the transport, this is for them, but this also leads to the intermediate signaling. On the other hand, this receptor can cross the barrier. All those receptors which can cross the barrier, they also come here, meet a receptor, and then all of this starts this mitotic activators. These mitotic activators are activated, which in turn start the transcription factors. When transcription factors are activated, what do? We all know they start the synthesis of the DNA. So this is a normal mechanism. So any proto-oncogene go mutated either at the ligand site, either at the receptor site, either at the signaling, like intermediate signaling site, either at nuclear site. All of these can lead to hypercellularity. All of these can lead to cancerous cell accumulation, because anything goes wrong in this pathway, this will lead to multiple cells. Those cells which are not even required, and that forms the cancerous cells. That forms a bunch of cells which we call cancer. And that growth is unregulated. As you can see, this is a steroid hormone receptor which is present inside, and in the nucleus, all of this mitotic activators and transcription factors are working, and DNA synthesis is happening. Next on, we talked about these ligands, we talked about these receptors. So in the proliferation mechanism, we have this balance between receptors and ligands, but receptors are far more in quantity. As you can see, the ligands are androgens and estrogens, and SIS and HSG receptor ligands are present. On the other side, on the receptor side, we have KIT receptors, we have ERBB2 receptors, and we have EGFR receptors. So the balance lies on the receptor side. Next on, we have proliferation mechanism. We just talked about anything can go wrong at any place, at any level, at any proto-oncogenic level. So if any mutation goes through proto-oncogene, it converts the proto-oncogene into oncogene. So if receptor binding transduction molecules are affected or mutated in some way, what will happen? That receptor site is sending more and more signal. That receptor site is not letting go of the ligand. Anything of these can happen, and this will give more and more intermediate signaling, more and more cell will proliferate, and we will have hypercellularity, and this will lead to cancerous cell mass in the body. It includes SRC, LCK, and yes. On the other hand, if the problem is with intermediate signaling, if mutation occurred in the intermediate signaling, and the intermediate signaling is, for example, if a ligand gets attached to a receptor site, it should send one signal. But if there's a pathology of intermediate signaling, it will keep sending on that signal, and the nucleus will think that more and more cells are required. It will keep proliferating, and it will lead to neoplasia. And it includes B, RAF, and MAPKs. Next on, we have nuclear receptors. Ligands got attached to receptors. Intermediate signaling helped it reach the nucleus. Now the nuclear receptors are there. Maybe the pathology lies within the nuclear receptors. They are perceiving the intermediate signaling totally wrong, and that somehow is affecting the whole mechanism. That somehow will perceive more and more signals, and it will keep multiplying, it will keep dividing until a cancerous mass is produced, a neoplasia has developed. It includes PPARs, androgens, and estrogens. Next on we have transcription factors. Maybe the problem lies within the transcription factors. That can also lead to neoplasia. Examples are MYC, FOS, JUN, and MYB. Next on we have growth factor related signaling and oncogenesis, meaning what growth factor will have the impact on signaling and oncogenesis. Those proteins that drive the cell proliferation can either attach to ligand. In the ligand, they can either, in the mutation, they can either be amplified. The signals can be amplified, and when the signals are amplified, we know the term amplification. What does amplification mean? That part of the DNA will be overproduced, and that will lead to amplification. And this is a type of mutation. It will lead to neoplasia one way or another, and HST and FGF3 lies within this boundary. Next on we have mutation in the form of depression of a gene. If a part of the gene is depressed, we see that when a part of the gene or when a part of the chromosome is depressed, it's not functioning. It's not functioning normally in any way. So that's where the SIS lies. Next on, it can either be intracellular signaling intermediate. The pathology may lie within this, and it includes point mutation in which one part of the genome, one point of the genome will be mutated, and it will lead to some effects which will be causing the neoplasia afterwards. And they are RAS, BRAF, and SRC. And next on, we have translocation in which one part of the genome will be translocated with another part of the genome, and ABL lies within this boundary. Next on, we have signal transducing receptors. First of all, we talked about all these receptors. We talked about all their mutations. We talked about MYC is going to be either amplified or translocated. So these are actually those receptors which are which are transducing the signals. So MYC will, in the mutation, will either be amplification or actually it's going to be either translocation or amplification. Next on, we have FOS, which includes only amplification. There's no translocation within it, lies within it. Next on, we have MYB, which also includes only point mutation. Next on, we have REL, which is amplification and point mutation. Both of them mutations are involved in this. And at the end of it, we have ETS, which acts via the translocation, which one part of the genome will be shifted to another part of the genome. That's why it's called translocation. So what's the RTK activity? The point is, these receptors we have studied, how will we know that which of these receptors are functioning properly? We have KIT, which is activating, the function of which is activating the point mutation. Next on, we have HER2 or new receptors, which will be, the function of which will be amplification. Next on, we have EGFR, which will include mutations and amplifications. Next on, we have MET receptors or MET receptors, which will include translocation. Then we have RHD, which will include point mutation and translocation. So in this part, in this KIT receptors, point mutations will be activated. Those mutations which are happening at a specific point, they will be activated. Whether they were functioning before or not, now they're activated. In amplification, more and more cells, more and more parts of that area will be produced. In mutations and amplification, it's being mutated and multiple copies of it are being produced. In translocation, it has been translocated with the other part. Maybe that part was non-active, now that's active, and this part was active and now it's non-active. Then we have point mutation, and a point is mutated and then translocated. It's not the same, but it's been translocated with the other one. So the activity depends on where it has been translocated. So in this lecture, we learned about all the different kinds of cellular activities. We talked about what cell cycle is, what's the resting phase, what's the G0 phase, how these phases coordinate with each other, what are the checkpoints of the cellular stage, that at which point the cellular stage will stop, will reconsider its going forward move, and then move forward. We talked about S phase in which genesis will be happening. We talked about G2 phase in which the cell will be enlarging. We talked about the mitotic phase in which cell will be going through mitosis. Then we talked about how cyclins and cyclin-dependent kinases are going to affect it. We talked about these are the positive regulators of the cell cycle. We talked about what are the negative regulators of the cell cycle. We talked about P53. We talked about all the other kinds of negative regulators of the cell cycle, how they affect them. We talked about how P53 is regulated. We talked about how different kind of inhibitors like retinoblastoma family is being affected, how retinoblastoma affects the process of DNA synthesis. All of this combined in one paragraph is that this is the whole basis of developing a tumor. All of us are made of cell. All of us are made up of cells. Very minute cells. If they're cancerous, they'll lead to tissue, a cancerous tissue. Then it will lead to neoplasia of the organs because it's moving forward. Those little cells which are moving away from the location they were born and moving into organs, they'll affect the organs. Then the organs will be affected, and as a whole, person will be affected. So all of this starts at the very basic level of cell and at the very basic level of cell cycle. So that explains how important the cell cycle is, how important the regulators of the cell cycles are, and how inhibitory regulators of the cell cycle are very important for us to watch in order to avoid neoplasia. Thank you for watching.